In this tutorial, I am going to show you how you can use the Galaxy platform to perform genome assembly using the tool SPADEs. So for this tutorial, in order for you to follow, you need to make sure you have an internet connection and you should have an internet browser. And you should also make sure that you have created a Galaxy account. So if you don't have an account, just visit this page here. I will leave the link in the description box. Visit the page, then register. And then once you have logged in successfully, you can follow this tutorial. So with this tutorial, what you are going to do is to first download an example data set, and then we are going to upload that data set to the Galaxy platform, and then we are going to trim the data set. We are going to use fastqc, so we are going to trim first, and then we are going to perform the novo assembly using the tool SPADEs. So that's what we are going to do in this tutorial. So let's proceed. Okay, so first we are going to create a new history. So come to the right side, check the history section, and then click on the plus button. So click it, and then a new history will be created for you. So it will be named as unnamed history, so we can edit it. So I'll edit it and I'll say genome assembly. So consider this as you doing a new project, and so you have to give it a name. At Galaxy gives it a name as history. Okay, so history is more like activities you are performing, but it can be a project, so that should be fine. So I will name it like this, and then I will save it. And so that means that if I check my histories, I can select genome assembly, and I will know that the data sets that have been generated in this particular history, that's a genome assembly. So it helps you to track your activities on the platform, basically. So that's about it. So now whatever we are going to do will be saved in this history. So we will start by uploading the example data. So the example data can be found here. I will leave the link to this page in the description box, as well as the publication of this data. I have used this data a couple of times as well. So let's proceed. So if you also want to learn how to use the SPADEs tool on the command line, I have a tutorial that shows that, so you can watch that. The link to that video is in the description, but you can also click this link right here. Okay, so let's get the data. So this is the data. We are using this EBI, and then this one as well. So we have read 1, read 2, that's R1, R2. We are dealing with paired and reads. So make sure you get this data onto your PC. Okay, so that's about it. I already have mine downloaded, so I will just show you. So this is the data that we are going to use. So we are going to upload this to the Galaxy server. So let's go back to the Galaxy server. So on the Galaxy server, just click on upload data. And then when you are here, we have regular, composite, collection, rule based. Okay, so these are the various options available. Now with the regular, it helps us to upload files from our PC. Okay, so if let's say you have the data on your PC, you can just click on the choose local files. So these are my files. Okay, I have the R1, R2. So I can select both and then click on open. Okay, and after that I can start. So you can also use the paste and then fetch data. So if you click on paste and fetch, you can give the name, okay, of the file, the name that you want the file to be labeled, and then you can paste the download link. So let's just try. Okay, let's try the same data set here. So let's look at how you can do that. So if you are here, just copy this link, and then you can paste it here. Okay, so let's just give it a different name. So let's just say PR2. Let's just give it this name. Okay, it's basically the same file, but I just want to show you how to upload from your PC and how to fetch from the internet. So this is for this one, and then we can go to paste fetch data again, and then select the other one here. And then place it also here. And we can give it a name. So let's say PR1. Okay, so you can do it this way. So I've shown you how to upload data from your PC and also how to get the data from the web, okay, by entering the download link. So that's about it. So let's upload these data sets. So we can click on Start. And then once we click on Start, Galaxy will upload the files. And then if the files are not on your PC, like the PR2 and PR1, then Galaxy will download them onto the server for you. So let's wait for the download to complete. So everything has been done, so we can close this one. Okay, so the PR1 and then PR2 are being fetched because these ones we are using the link, so Galaxy will download them to your account for you. So those ones are currently loading, but these ones we uploaded from our local PC and they are successful. So okay, so the files have been fetched for us. Okay, so we are going to perform the activity. So let's proceed. And I repeat that these and these are the same, so you don't have to use all of them. So you can choose to use these two or these two, but I prefer we use these two because we uploaded it from our PC. So that's about it, but the options are yours to decide. So let's proceed. Okay, we are now going to perform the exercise. So first of all, let's perform quality control to access the data. So I'll use fastqc. So I'll say fastqc. And then I'll select it here. So here we can select the data that we want fastqc to analyze. So if you want multiple data sets, you can just click this one here and then select the ones that we want. So we are going to use this and then this. These are the two that we are going to use. So that's it. We are going to leave all other settings as they are. So we will execute. So once we click on the execute button, fastqc will start the analysis and then it will generate reports for us. So let's wait whilst the reports are being generated. And sometimes too, the job will complete, but then the colors here may not change. So you can just refresh your page here and then see what comes up. But before we also proceed, let me also say that when you execute a job, you have some statements here telling you the output files. Okay, so with the fastqc report, it tells you that these are the 4 outputs that will be generated. We have a web page and then the raw data for each of the files that we selected. So we have 4 files, okay, 2 for each of the files. And so that's about it. So let's refresh the page. Okay, so it seems fastqc is still running. I'm not sure. I think it's probably queued at the moment. Let me just check here. I'll just check the status to see what's happening. Okay, yeah, it says this job is waiting to run. So that's why it's not colored at the moment. Okay, so I think it's the same for this and then this and then the other one as well. So we have to wait. So this sometimes happens. Okay, so depend on the workload of the Galaxy platform, some jobs may be queued. And if there's a free slot, those jobs are pushed there and then they're executed. So if you see this, you just have to be patient and then wait for Galaxy to run the job. Okay, so the fastqc jobs have finished running, and that is why we see the output files being highlighted in green. So I'll download just the web pages because these are the ones that have the reports. So I just check. This is data 2. So this is the web page for the R2, the reverse strand. So I will download it by clicking it, and then I'll come to this button here, this save button. I'll just click it, and then I'll save it on my PC. So I'm saving it here. I'll do same for the forward, which is here. That's data 1, and then I'll just save it. Okay, yeah, so I'll now go and then just explore those files. So these are the files. They come as zipped. And so what I'll do is to extract the content. So I'll open, let me just repeat again. So I'm going to extract the contents of each of these files. I want just the HTML, the one which has the fastqc report. So I'll open for the forward, which is data 1. And then I'll extract this one, p7741 underscore r1 fastq.gz.fastqc.html. This is what I'll extract. I'll do same for the reverse strand. So I'll extract this one. Okay, so I have both of them open. So these are HTML files, so we can open with our internet browser. So I'll open both with my internet browser. So I'll start with this, and I'll start with this. Now let's check the reports out on our browser. Okay, so these are the reports. This is for R1, that's the forward, and then this for R2, the reverse. So let's look at the reports here. I'll focus on the per base sequence quality. So if you check the per base sequence quality, you see that for the forward, we have all of them, almost all of them. I mean the bases, okay, the quality scores across all bases, so they are very high. So this indicates that the reads are of high quality because they are all within this region where we have the green color. So that means everything is okay. But we have just this one entering into this region here. So that's about it. Now let's look at R2. Let's look at the per base sequence quality. If you check it compared to R1, we have this observation where some of the bases, okay, they have low quality. So you can see that some of the box and whisker plots come to this side here, which indicates low quality, and we also have some here. Okay, this observation is not surprising. For Illumina reads, the quality normally drops at the end. And that is why we have this observation. So what we can do is to perform quality trimming. We will do this to remove the low quality ends of the reads. So that's what we are going to do next. So we will go back to Galaxy. So we are going to trim the reads. So let's check the reads that we are going to trim. We are going to trim these ones, the P7741R2 and then P7741R1. Okay, so we are going to trim, and then the trimmed reads are what we are going to use for the de novo assembly. So let's trim. So for the trim, we are going to use circle. So I will search for circle here, and then it comes up. So I'll just select it, and then when I come to this side where we have single end or paired end reads, I'll select paired end, two separate input files, And then when I come here, it's asking for paired end forward strand fastqc reads. So I'll select the P7741 underscore R1. And then when you come to the reverse strand, P7741 underscore R2. I will leave everything as it is. With the quality threshold, you can increase it as well, but I will leave it. Length, you can increase, but I'll leave it with the default settings. And then I'll execute this. So I'll click on execute, and then trimming will start. So let's do that. So when you click on execute, you have some information here telling you the output files. So these are the names that will be given to the output files. Okay, so that's about it. Now you can see that the jobs are running. So we have to wait for SQL to complete its work. Okay, trimming has been done. And so we can see the output files here. We are going to rename the output files. I am interested in the forward and then the reverse strands, okay, not the singletons. Okay, so let me just explain. So what SQL does is that it performs the trimming and produces three outputs. So we have the paired end forward strands, and then the paired end reverse strand, and we also have singletons. So the singletons are reads that do not have made pairs. Okay, so singletons will come from both forward and the reverse. So if you have reads in the forward without made pairs in the reverse, that is a singleton. So it's push here. If you have reads in the reverse without made pairs in the forward, it's also push the singleton. So that's about it. But I am interested in the forward and the reverse strands. Okay, so that's what I'll use for the genome assembly. So I'll first rename my files here because I usually have a naming convention. It helps me to track whatever I'm doing, and it helps me to organize my files. So I'll rename I'll rename both, but I'll use these two. So I'll rename this one. This is the paired end forward strand. So I'll click on edit, and then I'll say P7741. Let me do it again. I'll say P7741 underscore trimmed underscore R1. Okay, I will name this one. And then I'll also name the paired and reverse. So that one I will say P7741 underscore trimmed underscore R2. And then I will save it. Let's check to make sure that we have the same names. Okay, the same base name, so it's fine. Now with this output files, you can also download. So when you click it, you can come to the save button and then just download it, just as we did with the fastqc report. But I'll not download. I'll just proceed with the genome assembly. Okay, so now I'll search for SPADEs because it's time for genome assembly. I'll search for SPADEs, and I have SPADEs genome assembler for genomes of regular and then single cell projects. So that's what I'll select. And then after that, I'll have this window. So what's the operation mode? And there are a number of operation modes that we can use, but I'll maintain the first one, which is assembly and then error correction. And then when I come here, single or paired then reads, I'll select paired and individual data sets. And then when I come here, I'm supposed to choose the data that I want. So I'll just come to this side and I'll select the ones that are of interest to me. So let me do that now. So here it says multiple data sets, and here it says data set collection. Okay, I'll also use the browse feature here. So I'll come to the right side here. I'll select the browse feature to select the forward reads. So I'll select just this one here, this one. And then when I come to the reverse, I'll select the R2. So these are the files that I'm going to use. Now with the type of paired reads, the default is what I'll use. Orientation, I'll leave everything there. And then when you come here, you can select some options here. Okay, so we have isolate. It says it's highly recommended for high coverage isolated multi-cell data. We also have careful, which tries to reduce the number of mismatches. So it's recommended for small genomes. The data we are using is Mycobacterium ostrans isolate, which is small. So I'll use the careful flag here. And then the others, I will leave them as they are. That's about it. So I'll just execute. Please know that whatever I'm doing is just an introduction. So you can always read the documentation for SPADEs and see if there are parameters here that suit your needs, and then you can select them. Okay, you can select or deselect them. So take notes. So I'll just click on execute. And then SPADEs will start its activity. And so here it also says that 4 outputs will be generated. So we have the assembly graph for scaffolds, contigs, and then scaffolds. Okay, so these are the files that will be generated. So the assembly graph can be visualized using tools like Bandage. We also have contigs and scaffolds. With the SPADEs outputs, if you check the documentation, I checked myself, and then it's recommended to use a scaffold for the downstream analysis. So that's what I recommend you also use. So once you have your genome assembly done, you have the scaffolds. Okay, so let's wait whilst genome assembly is being done. Okay, genome assembly has been done, and that is why the backgrounds of these 4 files are now green. So from the parameter that we selected, 4 files have been generated. We have scaffolds and then the contigs. We also have the assembly graph. Okay, and the assembly graph files can be visualized using Bandage. Scaffolds and then contigs are fastqc files. They contain sequences. From the SPADEs online documentation, it is recommended to use the scaffold file for downstream analysis. And so what we are going to do is to just take a peek into this file. So I'll click it, and then I'll see this summary here. It says 2,437 sequences. You can download this file. You can also take a peek here. So that's what I'm going to do. I'm going to look at one of the sequences here, which is this one. So you'll find the sequence name. You also find the length. This is the length itself. And then you also have the coverage. Now the name, and then the length, and then the coverage for each sequence, okay, it can be stored in a different file. Okay, so we can actually specify some file names so that Galaxy will generate those files. So those files are called start files. So the start files, we will have a start file for contigs. We also have a scaffolds start file. Okay, so if you had specified those names, then we would have had our start files generated for us. So let me show you how to do that. So if you open your SPADEs tool, before you run it, if let's say you are here, you have selected your data, you've selected your pipeline options. When you come to select optional output files, you have these 4, and then you can click and then select the config start file. You can click again and then select the scaffold start file, and then you can execute. So when you execute, these 2 files, starts for contigs and then start for scaffolds, will be generated in addition to the original 4 files here, and then you can check the coverage information for all the sequences. So you can do that. Now after genome assembly, you can perform quality control. You can use tools like Quast. So Quast performs some analysis, and then you can use that to kind of assess the quality of your assembly. So you have N50, you also have misassemblies. There are a number of things you can do as well. There are a lot of information you can get from the Quast analysis. So I will encourage you to check that tool out. Aside that, you can also perform polishing. So polishing helps to improve the assembly. So you can use tools like Pilon when you are dealing with Illumina reads. So for this data that we use, it's paired end Illumina reads. So we can use Pilon to polish the assembly that we have generated. Please take note that scaffolds and then contigs, they are both draft sequences. Okay, so that's one thing you have to note. Now the SPADEs documentation recommends we use scaffolds for the dynamic analysis. So after assembly, let's say you have performed your QC, you have performed your polishing, and then you can actually perform genome annotation as well after you have done the polishing. Or even if you have not done the polishing, you can perform the genome annotation on the scaffolds that fastqc file. So you can use tools like Proca, you can use tools like PGAP, you can use tools like Rust, etc. So there are a couple of tools available for genome annotation. So you can use those ones to annotate your data. Okay, I've covered some tutorials on that, and I've also covered one tutorial where I use Galaxy to perform genome annotation. So you can watch that video as well. The link to that video is in the description box. You can also use the link right here to watch that video. So that's about it. So you can also call variants after genome assembly has been done. There are a number of things you can do after genome assembly has been done, but it depends on the questions that you want to answer. When I say question, I mean the biological question that you want to answer. It depends on that, and then you can use the appropriate methods or appropriate tools to try and get more insights, okay, from your data. So that's about it. So I also encourage you to watch the Galaxy tutorials that are in this playlist. This will help you to get more experience when it comes to using the Galaxy platform.